Yep. Welcome, everybody. My name is Blakely Nixon. I am the Communications and Events Coordinator at the Validate Network. And today I'm very, very excited to introduce Dr. Lydia Mosi. Lydia is a Validate Network investigator and associate professor at the University of Ghana. Lydia brings today some expertise in Buruli ulcer, diagnostic tool development, and the integration of neglected tropical skin disease management. Lydia and her team use a One Health approach to investigate how the environments of animals and humans overlap and drive M. ulcerans transmission. In the seminar today, Dr. Mosi will present 2 decades of environmental laboratory research on Buruli ulcer, and this is a fascinating topic, and it's one we haven't covered much before, so I'm very, very excited to hear everything you have to say today. So thank you. So I'm going to hand over to Lydia. Thank you.
Thank you, Blakely, and good afternoon to all. I appreciate the fact that you've taken time off of your schedules to join today's seminar. I'm going to go ahead and share my screen. And Blakely, if you will, confirm if you can see the screens in slideshow.
I can see the screen and it's in slideshow. If anybody else has any trouble seeing it, let me know.
Fantastic. All right, so I've already been introduced. The only other piece of information I would add is that I am at the University of Ghana in the Department of Biochemistry, Cell and Molecular Biology, which also has a center known as the West African Center for Cell Biology of Infectious Pathogens. So I have to say viewer discretion is advised right now because some of the images that I'm going to be showing very early on in this presentation are a bit disturbing. I'll start by first introducing the culprit of these disturbing images, Mycobacterium ulcerans. It is an acid-fast microbe. It's historically known as an environmental microbe, and it is slow growing. It takes about 4 to 6 weeks to form a colony on an agar plate, and it has a very restricted growth temperature between 28 and 32 degrees Celsius. Typically, the bacteria tends to form cords in growth when you look at it under a microscope, and it's simply because of the waxy cell wall, which is populated by a lot of lipids that are characteristic of mycobacterial species. Now, globally, if you ask where you can find Buruli ulcer disease, which is caused by Mycobacterium ulcerans, Africa comes up very strongly. Quite a few countries in Africa have been reported as having Buruli ulcer cases. And Australia is another country which is endemic for Buruli ulcer. There are certain countries in East Asia that have also reported occasional cases, but typically within the endemic regions in Africa and Australia, you have as many as 300 cases being reported in a year. Currently, it is considered the 3rd most important mycobacterial disease after tuberculosis and leprosy. And it's been shown through several studies that there's a high correlation between where the disease occurs and some sort of environmental disturbance, such as turnover of soil or flooding. When we zoom in closely in Africa, West Africa is primarily affected. The first case of Buruli ulcer on the continent was reported in Uganda, but since then, very few cases have been reported there. However, you have most of the cases being reported in Benin, in Cameroon, in Ghana, and in Cote d'Ivoire. And this graph here is a summative information of cases that have been reported to the WHO between 2013 and 2023. As you can see, Ghana and Cote d'Ivoire come up strongly, and it's pretty much because most of the research efforts have been focused in these countries. Having said that, we think that if you undertake active case surveillance in other countries that are here on this graph, you'd realize that there are more cases than have been reported. It is actually one of the difficulties or challenges we have with understanding the epidemiology of Buruli ulcer, simply because of the underreporting that exists in the field. Now, this is where we have some of the images, so please protect your eyes if you are easily disturbed. The disease starts as a nodule, which is actually palpable. You can move it around. And sometimes, instead of a nodule, there is a plaque which forms over a large area of the body. Sometimes also, the disease starts as an edema or a swelling of a limb. And in the unlikely case of treatments being sought late, it can develop into full-blown ulcers. A hallmark of Mycobacterium ulcerans infection or Buruli ulcer disease is that in all of these stages, there is absolutely no pain associated with the infection. Now, when you perform laboratory studies using different models, for instance, in a mouse model, depending on the inoculum or the dose which is delivered into the animal model, there is very early on some sort of intracellular stage of the bacteria where immune cells are capable of taking up the microbe and trying to clear them. In about 24 hours, again, depending on the dose which is delivered in the inoculum or the model of the animal, you realize that there is some sort of clearing of the infection. However, when we look at human infections, what is clearly evident in stained or histopathological tissues of the infection is extracellular occurrence of the bacteria and an area of necrosis and apoptosis in the region where you can find the bacteria. And it is very focal or localized during the infection. It does not go systemic in humans. Now, the culprit for the pathology that is observed during infection has been identified to be mycolactone. Mycolactone is a lipid toxin, which is comprised of a lactone core and a variable side chain. And I'll explain why it's variable in a few seconds as we go on. But this molecule is contained or is produced by a huge plasmid, which is found within the Mycobacterium ulcerans bacterium. This plasmid primarily has three main genes which produce the core lactone and the side chain, and some of the other genes add accessory groups to the side chain and to the core of the molecule. Now, I know this slide is busy, but I'm going to try and talk through it to appreciate the extent of the pathogenesis of mycolactone. So by itself, without the bacterium, mycolactone is capable of inducing an ulcer, which is akin to Buruli ulcer lesions. George et al. In 1999 was able to demonstrate this very clearly and also to prove that it was not a protein, but actually a lipid toxin. Several studies out of N's group in Japan, as well as other researchers, have also identified that the toxin is capable of killing nerve cells, and that's what has been found to be responsible for the painlessness of the lesions. A large group of researchers from UK, University of Surrey, and in Belgium, from Caroline de Mangel's group and Rachel Simmons' group in Surrey, have also shown the immunosuppressive capabilities of mycolactone. And whether it's delivered in low doses or in high doses, mycolactone is capable of inhibiting T-cell production, inhibiting the production of co-stimulatory molecules by dendritic cells, and also inhibiting the production of other cytokine molecules. In all of this as well, mycolactone has been shown to be responsible for the coagulative necrosis that are found in ulcers. And Fred Saffo and other researchers have been able to show this in very beautiful experiments. Another large group of scientists from Pam Small's group and other groups have also shown the cytotoxicity associated with mycolactone on all cell types except red blood cells and platelets. And mycolactone is capable of inducing apoptosis and necrosis and arresting the cell cycle, among other things. Having said all of this, it is very evident that mycolactone is absolutely crucial and necessary for the pathogenesis of Buruli ulcer. How is the infection diagnosed? So whether it's presented as an ulcer or as a nodule, swabs can be taken from the open lesions, or fine needle aspirates can also be obtained from the nodules, as well as tissue biopsies from the ulcers. And all of these are subjected to any of these methods, which I'm going to uncover in just a second. So historically, microscopy has been used in the diagnosis of Buruli ulcer, especially in resource-poor areas, where the classical detection of extracellular bacilli that have been stained acid positive are deemed a positive diagnosis for Buruli ulcer. Occasionally as well, some advanced labs have been able to use histology to observe the characteristic necrotic and apoptosis associated with the infection and classify the infection as Buruli ulcer. Culture is also used. However, it is very, it's the most specific, but it is not a very successful diagnostic tool to use because of the duration it takes for colonies to show up. By then, the ulcers would have extended beyond repair. This means that polymerase chain reaction or molecular detection of the presence of the bacteria is the gold standard currently, as suggested by WHO. And there are several markers which are used to identify the presence of the bacteria, including some insertion sequences and other genes which are responsible for making the core or the side chain of mycolactone, the toxin involved in pathogenesis. So treatment, Buruli ulcer can be treated, especially if it is diagnosed and reported early. Most of the time, surgical excision of the lesions is conducted into healthy tissue, followed by skin grafting. Alternatively, if the lesions are very small, a combination of antibiotics is very useful in treating the infection. Rifampicin with streptomycin or clarithromycin or moxifloxacin are all possible treatment regimens for Buruli ulcer. I must state that the durations of the antibiotic therapy are quite long, lasting about 16 weeks to clear the infection. However, in all of this, one hallmark of Buruli ulcer that remains even after the lesions have resolved is the characteristic scar tissue formation that occurs in people who have been affected. And if this happens in a limb or in a joint of a limb, you can imagine the loss of mobility and how this affects both their ability to conduct work and also the stigma which can come with having all these sort of scar tissues on your body. Now, away from the disease and its presentation, when we take a look at where M. ulcerans is placed in relation to other mycobacterial species, you realize that it is very closely linked. Looking at the 16S rRNA gene, you realize that it's very closely linked to Mycobacterium ulcerans and Mycobacterium tuberculosis complex. MTB is an intracellular infection or produces an intracellular infection. However, M. ulcerans produces an extracellular infection in man. M. marinum can produce both an intra and extracellular infection in man and in other organisms like fishes. Now, a couple of years ago, other species of mycobacteria were identified in other organisms. So M. liflandii were identified in captive frogs, which were kept in an aquarium, and these were able to produce a systemic infection in frogs. M. pseudoschottii and M. marinum subspecies DL and CC were also identified very shortly in fish. The underlying factor between M. liflandii, M. pseudoschottii, and M. marinum subspecies DL and CC is the fact that they all produce mycolactone. They all have the toxin that is capable of producing mycolactone. And when you take a look at their 16S RNA phylogeny, you realize that they are very closely related to M. marinum and M. ulcerans, right here and here and here. So what makes is there a difference between the congeners of mycolactone that these fish and frog isolates make compared to the congener that is produced in M. ulcerans? The answer is yes. So what is common to all of these different mycobacterial species is the fact that they make the same core molecule. So they have on the plasmid the machinery that is capable of making the core lactone molecule. However, the variations exist in the side chains of the molecule. And these variations are typically either a shortening of the side chain or differences in the accessory groups that are found on the side chain. Now, these variations go hand in hand with the level of virulence of the different congeners of mycolactone, with M. ulcerans having the most virulent variant of mycolactone. So with all of this information, what we know up until now is the fact that marinum and ulcerans are very closely related, with under 3% of their genome being different. So the theory to date is that M. marinum, through the evolutionary time frame, acquired the plasmid, which is responsible for making mycolactone, and that plasmid also acquired several insertion sequence elements, as well as the genome of Mycobacterium marinum, to result in the generation of the new strain Mycobacterium ulcerans and the other MPMs, or mycolactone-producing mycobacteria. The thing which is common in all of these mycolactone-producing mycobacteria is the fact that they all have mycolactone, albeit some have a difference in the nucleotides to result in the different congeners of mycolactone that we have. So when we ask ourselves, what is the transmission, or how is Mycobacterium ulcerans transmitted to man to result in Buruli ulcer infection? This is one of the biggest challenges that exist in the field or in the study of Buruli ulcer, because we do appreciate that there's a context in the genome reduction from M. marinum to result in M. ulcerans, but we don't understand how that plays a role in how it is transmitted to man, and also whether these other mycolactone-producing mycobacteria are capable of causing infections in humans. What we do know is that if you go into a typical endemic region where you have Mycobacterium ulcerans infection, you would find the bacteria everywhere. When I say find the bacteria, I mean you would find the DNA of the bacteria everywhere. It's in the water, it's in the soil, it's on biofilm, which is attached to the surface of plants. You can find it even on the surface of any organism, like an insect that frequents the water body. And some of the challenges which affect our ability to understand how it's transmitted is the fact that, yes, culture is the gold standard to prove that the bacterium is actively growing here, but it's really difficult to do that, bearing in mind the slow grow rate of the bacterium. Also, the current methods that we have available to detect the presence of the bacterium are limited to the presence or absence of DNA, and just that doesn't tell us that the bacterium is alive. Also, the presence of these mycolactone-producing mycobacteria in these same endemic areas poses a challenge for us to be able to diagnose whether it is truly emulsions or these MPMs. And the fact that you can find the DNA everywhere makes it a very convoluted web, which again makes it difficult for us to pinpoint the exact place or reservoir where the bacterium is surviving or multiplying. Finally, I do have to state the role of small animals who live or frequent these kinds of endemic regions in the transmission. And this is still an area for exploration in studying Buruli ulcer transmission. So there have been some working hypotheses that have been proposed to date. So we do understand with different studies, some coming out of my group, that M. ulcerans during its course of evolution has adapted significantly to be different from a strict environmental microbe, in the sense that it is probably obtaining its nutrients from a host or a niche where these nutrients are very much available, and it is adapting to an area where there's limited oxygen and a lower pH. Some groups have been able to show that or associate non-mosquito aquatic invertebrates or insects in the transmission of Buruli ulcer. And most of my PhD work was actually trying to understand or uncover this. Other groups have also shown that if you have a topical, or should I say, a sore or an open wound on the surface of your skin, it's enough to produce an infection of M. ulcerans, of Buruli ulcer, if M. ulcerans is deposited on the surface of the broken skin. Other groups, including people from my group, have also tried to associate or understand the association of small mammals in the transmission of Buruli ulcer to see if they can serve as reservoirs. But most recently, the Australian group have been able to show through very meticulous years of experiment that in their context, mosquitoes is the transmissible host for Buruli ulcer in Australia at least. Now, if you go to the environment, like I alluded to earlier on, and you try to test all the samples that you can obtain in an endemic region, you will find M. ulcerans there. And several of the markers that we use routinely include the ER and KR domains, which are all on the plasmid of the mycolactone, producing plasmid. And the insertion sequence is IS24 and IS2606, which appears several times, both in the genome and on the plasmid. Variable number tandem repeat typing has also been very useful in identifying or in differentiating between the strains that we find in these environmental isolates. But what is evident is that IS244 is the marker which most research groups use. Now, in our efforts to understand transmission, we have, in small silos, taking samples from only the ulcer, or we've taken only environmental samples and looked at them to see if you can find Mycobacterium ulcerans there, or we've taken only small mammals. But I think the best way to link all of these things up is if we attempt a One Health approach and formulate our studies to include looking in endemic regions for the presence of the bacteria in the human, in small mammals which live within the vicinity of the humans, and also in the environment, in the immediate environment of the humans. This way, we'll be able to understand if it's the same strains which are circulating and whether or not the infection is just purely opportunistic or really needs one of these reservoirs or hosts to keep producing an effective infection. I do have to underscore some of the challenges that we have in trying to understand transmission, particularly in West Africa. We have several slow-flowing water bodies, which is a classical risk factor of Buruli ulcer. We also have a lot of human activities in these water bodies, fishing, farming, swimming by children. So it serves as a risk factor as well. The fact that people come into contact with these water bodies is a risk factor. Also, the fact that cases are reported late, so most of the cases are seen in the 3rd stage or in category 3, where you have full-blown ulcers. Late diagnosis is a big issue, which affects our ability to understand transmission because we cannot link it with when the infection started. There's also the whole climate influence issue where we can't quite pinpoint a seasonal variation in when the incidences of Buruli ulcer rise and when they fall. And then finally, because of the stigma attached with the disease, people who even notice that they have a painless wound don't report to the hospital because they're afraid of being treated as outcasts within their communities. It is good to point out at this point that Buruli ulcer is a very rural disease in West Africa. You don't really find it in the urban areas, you find it in the rural areas where access to healthcare is really a problem. So I'm going to highlight very quickly 4 studies which we've done in the past to try and understand transmission. And the first one was between 2011 and 2015, where I worked as a postdoc at Sanchez-Suez de Research Scientifique in Côte d'Ivoire, in Prof. Bonfo's group, Basiru Bonfo's group. So my role in that as a postdoc here was to first establish a molecular biology lab and Buruli ulcer research at CSRS. And the whole aim was to understand if zoonotic transmission of M. ulcerans as well as other non-tuberculous mycobacteria existed between humans and small mammals. So the study was conducted in Ghana and in Côte d'Ivoire, which are countries which right next door to each other. So human samples, animal samples, environmental samples were obtained from endemic regions in Ghana and in Côte d'Ivoire. And these samples were tested using molecular methods to determine the presence of M. ulcerans as well as other non-tuberculous mycobacteria, all in a One Health approach. So the main objective was to molecularly source track M. ulcerans to help us understand Buruli ulcer transmission, and also to perform socio-anthropological studies to identify possible risk factors of transmission. So the first data that came out, or most of the data, this is a lot of data summarized in one slide, but we tested environmental samples, and for the numbers that we looked at, Ghana had most of the positivities when we looked using IS2404 as the PCR marker. And when we used mycobacterial 16S ribosomal DNA as the marker, Côte d'Ivoire came up more strongly positive. When we looked at the human cases that we were able to sample or obtained, we were able to conduct and confirm 98 case positivity in Ghana versus 94 in Côte d'Ivoire. And then we also trapped quite a few small mammals. Most of them were rats, and these traps were placed in the homes and kitchens of those who consented to be in this study. And we used Sherman traps, so we were limited to trapping mice or rats and not any other type of rodents. And for the first time in West Africa, we were able to show PCR positivity of M. ulcerans DNA in both Ghana and in Côte d'Ivoire. And we summarized all of this data on a geospatial map, where the rectangular callouts are VNTR profiles of the isolates of the strains of samples that tested positive in the environment, and the circular callouts are sample positivity in the human and in the human people who the people that we tested in the study. Now, what is very evident in this is genotype Y. The NTR profile genotype Y is the profile that was common, or should I say the most common, in both the environment and in humans in the areas that we sampled. And this is a classical example of a map that we were able to draw for Ghana. There's another one for Côte d'Ivoire. And this shows how you can use GIS or geographical placement systems to understand infection or transmission or along a water body or along a geographical area. The red triangle is indicative of some of the areas where we found small mammals being positive. And so some of the lesions that we identified on the small mammals, we went on to sequence our PCR positives, and they came up with 99 and 98% respectively identical to M. ulcerans Aggie strain, which is the genome strain. Now, this work was able to train 4 students at the PhD and the master's level, and they all had publications from this work. But I think what's also important is the fact that we are able to build capacity with this kind of research that we do and train more people on using molecular methods in diagnosing neglected tropical diseases. Now, the second piece of work that I want to show you is from Bright Azuma, who was my master's student, and he wanted to look at the involvement of Acanthamoeba polyphagia in the transmission of Buruli ulcer. So his research question was, can M. ulcerans survive within Acanthamoeba polyphagia, and does this virulence increase? Does it increase virulence? So in other words, if you passage M. ulcerans through an amoeba, and you use that as an inoculum for an animal model, would you have increased virulence or not? And he was able to show that, yes, indeed, M. ulcerans could infect amoeba. The amoeba didn't die, depending on the dose of inoculum that was delivered. And we also used red fluorescent protein tagged M. ulcerans, which we use to track the viability of the M. ulcerans within the amoeba. And as you can see from the overlay of these images, during the course of the infection, the M. ulcerans were still alive and viable in the amoeba. But more importantly, Bright was able to show that the passaging of the M. ulcerans through the amoeba made them more virulent. And so mice that were infected with recovered strains from the amoeba produced an infection quicker, and this infection was had the pathology which was similar to Buruli ulcer lesions. The third piece of work that I am going to show is work which was done in collaboration with Rachel Simmons at the University of Surrey. And this is one of the studies which also received funding from Validate, the pump priming funding. And so thank you very much, Validate, for supporting the work that we're doing here. So this work, this study sought to understand the genetic diversity of mycolactone producing mycobacteria in Ghana and in Cote d'Ivoire. So again, we went into the field and collected environmental and human samples. As you can see, the collaboration team was huge because we involved hospitals that were the primary treatment points of Buruli ulcer in both Ghana and Cote d'Ivoire. And this work was done by Elizabeth Jemfi, a former PhD student, and Magdalene Dube, former master student in our group. And I'm going to show you some of their data, which they did using VNTR typing or profiling of the strains that they found. And on the left, I'm showing you the different types of profiles which were identified in environmental samples compared to on the right profiles which were identified in human samples. So as you can see, most of the environmental samples have more variations in their VNTR profiling, or should I say, we had more mycobacterial strains. And again, I'm quote unquote mycobacterial strains in the environment compared to humans. In other words, it's not all the environmental isolates or strains which are capable of producing human infections, at least in the areas that we looked. But what was evident again is the fact that profile D, which again types with genotype Y that I showed earlier on, which you see most in the environmental samples, are what are responsible for causing most of the human cases. And then using Oxford nanopore technology, we were able to we were able to sequence the genome of 3 of the isolates that we were successful in culturing. And this we are still working on. We want to confirm the genomes that we obtained using Oxford nanopore with Illumina sequencing, and this data would be published soon. Again, training, capacity building. It's the key to active case surveillance for neglected tropical diseases. Occasionally, we even have to procure vehicles to help with obtaining samples from the very rural and endemic areas where there's no access to cars to transport cases. But this training helps in obtaining community health volunteers who can identify a lesion as Buruli ulcer and help to report the infection so that treatment is sought early. The fourth piece of research that I want to show you is by a student, Kwabena Boateng, who is currently undertaking his PhD work in Rachel Simmons' group at the University of Surrey. So he worked on a study that sought to answer whether mycolactone was produced intracellularly within macrophages in that brief time that they were in a cell. And so he was able to infect macrophages and show in a time course that up until 24 hours, the bacteria remains intracellularly. But after that time point, the bacteria starts multiplying and killing the macrophages. But then he also showed this data with a live and dead stain to show that after 48 hours, the bacteria starts killing the macrophages, and then they multiply in number compared to M. smegmatis, which is another environmental mycobacterium isolate. And then he performed some qPCR experiments to show in real time that transcription, the regulation of mycolactone is not affected, does not happen at the transcriptional level, at least from the data that we had, and that the macrophages are able to, bacteria is able to reside in the macrophages up until 24 hours. And you can see that key accessory genes like the ER domain is differentially regulated at different time points, as you can see here. And then finally, some more work, but this time using bioinformatic modeling to try and understand the role of pseudogenes, which are plenty in the genome of Mycobacterium ulcerans, to see if they can help us understand how M. ulcerans is adapting to the current niche that it finds itself in, and to also understand the evolutionary differences that exist across the different strains that have been identified in Africa. And so Edwin Chebafo, who was a master student in my group, was able to obtain sequenced genomes of M. ulcerans. So these are human isolates that were obtained from different African countries. And he developed a bioinformatic pipeline, which he used to appreciate the fact that there are two circulating lineages of the bacterium in Africa. And these lineages can indicate the level of virulence of the infection that is caused by the specific lineage. And also he was able to show the different mutations that existed in the pseudogenes across these lineages, and the different pathways or metabolic pathways within the bacterium that have been inactivated as a result of these genomic differences. At the end of the day, using the data that he generated, he was able to show an evolutionary track of M. ulcerans and observe the fact that it is adapting to an area that doesn't have a lot of competition, like a strict environmental microbe well. It's adapting to a niche where it is protected from direct sunlight or UV rays. It's also adapting to a niche that has low oxygen and has availability of nutrients for M. ulcerans. And it's also adapting to a niche that can support or that does not respond to the cytotoxic effects of mycolactone. So what is the takeaway from all of this information? Is M. ulcerans really everywhere you look? That's the key question. And if we are limited to just looking for the presence of the DNA, then yes, it is everywhere you look. So I think it is now important that if it is everywhere that we are looking, then we should start targeting our studies with a more One Health approach to try and pinpoint the key reservoirs where the bacterium is multiplying and help us to understand transmission. I think we also have to adopt a more standardized methodology in our approach to studying the environment where M. ulcerans is endemic, and also the kind of markers that we use in the testing for either case confirmation or presence absence of the bacteria, we need to have a more standardized methodology so that what a group is reporting in Ghana would be the same in a group that is being a group that is doing the work in Europe. Also, I think we need to go beyond looking for the DNA and start seeing if we can determine the signature of life or even the presence of RNA in the samples that we obtain, because that helps us narrow down to the actual niche where the bacterium is alive, viable, and actively multiplying. And I cannot underscore the importance of working in a transdisciplinary approach. So in devising or in designing our studies, we need to involve all manner of expertise, including microbiologists, anthropologists, herbalists, epidemiologists, and even the community in planning our studies to better understand transmission. Now, WHO has its primary focus currently in Buruli ulcer on trying to prevent and control infection. And I think that if we are able to block transmission, then we'll be able to have zero Buruli ulcer in the world. And with that, I'm really grateful to the different funders who have supported the work that we've done in our research group, and more recently to WHO and the Muela Malasela Mentorship Program for recognizing the work that I've been doing here in Ghana. And with that, I'm going to stop share and take any questions if there are any. Thank you.
Yeah. Thank you, Lydia. That was excellent. Tour de force. If you don't mind, I'm just going to post some information about Validate in the inbox. So we've got plenty of non-members. So Validate is a network of vaccine researchers working towards vaccines for mycobacteria, which includes Buruli ulcer, leishmaniasis, and Burkholderia pseudomeliae. And membership is free, and there are plenty, and I've just put lots of information in the chat, a couple of links in the chat. So with that in mind, does anybody have any questions for Lydia? You are more than welcome to type in the chat or just, yeah, Andrews, would you like to ask your question? You should be able to turn your mic on. Yeah. Andrews? There you go. Okay, nothing from Andrew. Belinda, did you have a question?
Yes, sorry, I couldn't work out how to put my hand up. Yeah, I just wanted to ask you, Lydia, for that first say, nice talk, thank you. The mycolactone producing mycobacteria that you found in the environment that you couldn't find in people, do you think that's because they're just not infectious to people, or because they're occupied in a niche that means they don't get transmitted?
I think the answer is both, because again, this is just DNA information. And I can't tell whether these are in they are in a state where they can be easily transmitted. I can't even tell whether they would adhere to the same transmission hypothesis that we have for M. ulcerans as we know. So it could be either option. It could be the fact that they are somewhere in the environment that is not readily available or in contact or can make direct contact with humans, or we just haven't looked deep enough to see whether they are infective, just like M. ulcerans.
Yeah, but it might give some clues as to what the niche is. Thank you.
Thank you.
Hi, David. Anon, did you have a question? Oh, Andrews? Oh, Andrews is coming now. I can't quite hear you.
Can I ask my question?
Oh, go ahead. Go ahead, Andrews.
Yeah, hello, Dr. Lydia.
Hi,
yes, I understand it's a neglected tropical disease, but in Ghana, we have so many villages, and I've not seen a situation where most villages are reporting this. but it seems specific to some specific villages. And you mentioned something like about 130 people, and then the DNA is everywhere. It beats my mind a little bit because it seems to me that the bacteria takes advantage of a few people with some with their immune system compromised.
Yeah.
Other than that, if you take a village or a household, do you see all the people in the household infected, or just one or two people?
Yeah. I think that you've just underscored the conundrum that we face in the world of transmission. So person-to-person transmission has not been reported. So you can have people within the same household. One person has Buruli ulcer, and the other people don't have Buruli ulcer. Having said this, we do appreciate the fact that there is a key immune component that we quite haven't uncovered. And when I do say that the bacterium is found everywhere, it means that if you go into a typical endemic region and you sample the classical water bodies that have been associated with transmission, you will find the bacterium in almost every organism type, even in detritus in these environments. Having said this, if you go into a non-endemic area, the story is not the same. You don't find emulsions there. So there has to be something in the environment where the cases are endemic or where the cases are occurring that supports the growth of emulsions directly in the environment. And that's what some of the work that we are doing is hoping to uncover. I hope I've answered your question.
Yes, I get you.
Thank you, Andres.
Thank you very much.
David, did you turn your camera on because you wanted to ask a question? David Anor? Maybe not. Okay. Does anybody have a question then for Lydia? Oh, there's a question in the chat from Sparsha Palin, who says, thank you for the talk. Does M. ulcerans have any specific specificity to children, or can it equally cause severe ulcerations in adults?
So at least in Africa, there hasn't been a clear-cut association with children. It can affect all age groups. We were just in Australia recently, and most of the cases in Australia are in adults. So again, it doesn't discriminate between whether you're a child or you're an adult. And this is a classical hallmark of an opportunistic infection. Hopefully that answers the question.
I think that does answer the question. So anybody else, you might have a question, you can turn on your mic and give us 30 seconds. You can turn on your mic, or you can type like Spasha did in the chat. Oh, there is one from Jonathan. Hi, Jonathan. How far are we from identifying the transmission route in West Africa using the One Health approach? That's from Jonathan Adeji.
I think you're very far away until we start working together. More hands on deck. Like I alluded to in my concluding slide, we need a lot more expertise from different disciplines to help us really use the One Health approach in understanding transmission. I did mention that most of the cases or most of the research that has been conducted up until now have happened in silos where people are only coming to collect human samples, or people are only collecting environmental samples and looking at those. But I think we need to start looking at all of the possible factors that have been named as risks of emulsions together collectively in one study. And it has to be a really large study where you have a classical endemic area or conditions that put you in a classical endemic area and a classical non-endemic area. And then you can compare all manner of things without having any biases. So you can compare the environment, you can compare the even the diets and lifestyles of people. We need that kind of approach, using One Health as our guide in order to really identify the transmission routes in West Africa.
I have a quick question, because this is Validate. We're a vaccine network, and broadly speaking, what is the prospects for vaccines? Is BCG effective in slowing this down, or so? In layman's terms, because I am a layman, what are the prospects for a vaccine for Buruli ulcer, and how does that play into the sort of work you've done?
Ooh, okay, so again, one of the things that is peculiar with Mycobacterium ulcerans and its closely related cousin Marinum is the fact that if we want to develop a vaccine, then it has to be able to detect that 1% difference that exists between M. marinum and M. ulcerans. That's already in itself is hard to do. People have tried to use the toxin, but then it's a lipid toxin, which is poorly, it's a poor immunogen, so you can't really use the lipid toxin. I know there are several groups that have tried to use aptamers to link certain groups in the toxin as a possible target. In its pathology as well, the SEC61 translocon has come up strongly as the main virulence mechanism of emulsions. But again, it can't be used as a vaccine candidate or even a treatment candidate because of the importance of its own role in normal cellular functions. So I know I'm sounding like the last Vader, but it's still work in progress, and hopefully we'll be able to identify a protein that the bacterium makes, or some signature of the bacteria in the host, which we can use as a vaccine candidate. So it's work in progress, and yeah.
Thank you. Excellently answered. I'm just going to give people just a few more seconds. If you have a question, you can type it. We've got you can type it into the chat, or you can just come on the mic. But just give you a few. Let's give them a few more seconds to get up the guts to ask a question. It can be a bit nerve-wracking. But other than that, I think I think that might be a nice time to wrap up. So I would like you all to somehow, either in the chat or just come on and wave, express appreciation for the excellent talk by Dr. Lydia Mosi in Ghana. So thank you very much. That was a brilliant talk. I'm sure so yeah, thank you very, very much. And so for you non-validate members, do check out the links I put into the chat and the ones I emailed you earlier, because this is the kind of thing that we do. And we do this in person with our annual meetings, and there's also other significant opportunities available through the Validate network. So do check that out. But one final thank you to Dr. Mosi. Thank you very much.
Thank you everyone for joining. Hi, Rach.
Hi, Rachel.